Our research work is interested in the application of pharmaceutical science research techniques to answering harm reduction questions. In particular, I'm interested in whether the equipment that gets given to injecting drug users from needle exchanges actually does reduce harm, and making sure it doesn't cause any harm in the process. Work has examined lots of different bits of paraphernalia, but part of our work has focused on filters. Filters are important because drug users use them to take out insoluble particles, lumps of drug and other material that could block the needle when they try and draw the liquid up into the syringe. And a blocked needle is potentially a wasted hit for a drug user, so the concern is not to have particles that could block the needle in the injection. This doubles up for us in terms of a health concern because particles can also block veins. They can travel around the body and they can cause lumps and other complications in internal organs. Commonly, they get lodged in the tiny little blood vessels in the lung, and people can present with symptoms which seem like a tumour, and on examination or biopsy it turns out that they have what's called granulomas in the lung. There's also concerns about particles causing clots, so being a sort of seed around which a clot forms and causing complications like deep vein thrombosis and pulmonary embolism. So our research has wanted to answer the question of, well, which filter takes out the most particles, and we want to take out the most and also the smallest, so right down to the smallest number. So in a sense, what you're left with is a solution that is not going to be the same as a pharmaceutically manufactured solution. It's going to always have way more particles than that, but it's somewhere towards the idea of being low particle count and small particle count, because the smaller the particles, in theory, the less the risk of harm. We've examined a whole range of filters, and we broadly split them into what we call makeshift filters, so the things we know drug users themselves will use: pieces of cigarette filter, pieces of cotton wool from a cotton bud, say, or other source of cotton wool, a filter from a hand rolling cigarette, bits of tissue, other things like that we've studied. And then we've compared these with what we call commercially available filters, not necessarily made for purpose, but filters nevertheless that can be fitted onto syringes. So this has included things like wheel filters that we use in laboratories to clear particles from solutions before they go through very sensitive analytical equipment, and filters like SteriFilt and similar filters that are made by companies and put into safer injecting kits. We've looked at effective filters on injections made with tablets, because tablets are very heavily contaminated with particles. They're not made to be injected, they're made to be swallowed, and when you crush them up, it creates a huge amount of particles. And we've also examined the effects on street drugs, specifically heroin. We've found that the commercially available filters that we have studied have taken out more particles than the makeshift filters, and the best of the makeshift filters was the cigarette filters, but nevertheless we've found better performance with the makeshift ones. There are some difficulties with their use. We've found that the syringe filters, the wheel filters, absorb a lot of liquid and can take away a lot of drug, and we've found things like SteriFilt, although they don't take out any drug, they're very negligible amounts of drug, and they only let through small particles. They're not that easy to use and take a bit of practice for somebody to get the hang of. We were interested in the development of harm reduction equipment and testing performance. Ideally, it would be nice to be able to compare performance in a clinical setting, do some sort of longitudinal study to look at the effects of different paraphernalia when used by users, but actually practically that would be virtually impossible to do, because we wouldn't be able to control the study very well. We couldn't reliably ask people or expect people to use one filter only ever for the long period of time you would need to actually examine the effect on their health. And it's pretty unlikely a study like that would get funded. So we have to rely on the laboratory information to make predictions about how these filters are likely to perform in practice.